my pleasure to introduce Keara Boss, who's coming to us or joining us today from Fort Collins in Colorado. Keara is an assistant professor at Colorado State University in the Department of Environmental and Radiological Health Sciences and the Flint Animal Cancer Center. She did her PhD in comparative biomedical sciences with a focus in cell biology through North Carolina State University and Duke University, and is going to speak to us today about a very interesting perspective or aspect on using radiation, and this is working with companion animal models. So Keara, it's a pleasure to have you join us, and I'm going to hand over to you, and hopefully you can share your slides, and we look forward to hearing from you.
Absolutely. Thank you very much for the introduction, and thank you so much for the invitation. Happy Friday. I'm really excited to be a part of this seminar series. So yeah, I'll be probably sharing some newer kind of alternative ideas for studying radiation in companion animal models. And let me share this. All right. And can you see it? Yes.
Yes.
Okay, great. And I'm going to minimize this. So I can't see anyone. I go kind of into the presentation mode kind of vortex here. So if anybody has questions or anything, please just speak up, because otherwise I probably won't see you. So yeah, I'm Keara Boss. I'm at Colorado State University, and I'm a veterinary radiation oncologist, as well as a radiation biologist. And I'm going to be talking to you about canine comparative oncology for radiation research. So I think, as we all understand and respect and recognize, laboratory research is essential for the foundation of scientific discoveries, mechanistic studies. But overall, there's become a more of awareness that developing effective cancer therapeutics is a relatively inefficient process when you consider the amount of time and energy and money that goes into benchtop research to bedside to actually moving into approved and integrated cancer treatments. So, you know, people are kind of looking for more alternative approaches to try to bridge this gap in translational cancer research. And so what I'll talk to you about today is canine translational models. And I'll say there's a great value in any lab animal model. So that includes kind of purpose-bred research dogs, which I do research in as well. But again, today, what I'm focusing on is research using companion dogs or people's pets who have been diagnosed with cancer and are undergoing treatment. So in terms of just some background and rationale for considering canine translational cancer research, dogs have a lot of very similar physiologic and biologic similarities to humans. There's about 6 million new cases of cancer that's diagnosed in dogs each year in the United States. And just as like a rough estimate, about a quarter of dogs in their lifetime will develop some kind of cancer. And dogs that are developing cancer do so, spontaneously. As a population, I know there's certain breeds, but considered to be outbred, more like people versus lab animal models that are very tightly regulated and bred according to certain strains. So you have a more degree of genetic diversity. Their age at diagnosis of cancer seems to be relatively similar to what's seen in people, as well as, just like people who develop cancer, dogs throughout their life may be diagnosed with cancer at the same time as having other comorbidities that need to be considered when undergoing treatment. And then for cancer research, more and more interest in immune responses. These dogs are immunocompetent. They have a natural immune system that they have been living their life in the same home and environment as their people that they have developed their cancer with. So another part of this is that dogs can go on to receive more advanced medical care when they're diagnosed with cancer. So there's a specialty field in veterinary medicine, veterinary oncologists. And so this includes the specialties of radiation oncology, like I said, that I'm a part of, but I work very closely with specialty vets who focus in medical oncology or surgical oncology. And so our patients go through the process of undergoing various diagnostic tests to either diagnose or then stage their cancer, and then through the phases of treatment. And just like people, unfortunately, we're monitoring them over time and are seeing when they have developed treatment resistance and cancer progression, as well as we're always monitoring our patients for any evidence of normal tissue toxicity that we also need to balance. Compared to laboratory animals, dogs are, or rodent-based animals, I'll say, they're a larger size. So when we're treating them in our hospital, a lot of the time we're using similar equipment that's used for human patients. For their cancer treatment, for many tumor types, they have a relatively similar course of disease. However, dogs, their lifespan is relatively short compared to a human. So we're able to evaluate experimental results, treatment outcomes on a shorter time frame than you can with a human clinical trial. So in terms of canine translational radiation research, so this isn't kind of a new thing. This has been ongoing for decades. There's a pretty extensive history of canine translational radiation research that actually was pioneered by Dr. Ed Gillette, who was faculty here at Colorado State and actually trained all of my mentors. He did a lot of work with his team evaluating radiation responses to both tumors and normal tissues in dogs. And the research went on to benefit both humans and dogs who are undergoing radiotherapy. And so now there's still more and more ongoing research utilizing canine comparative oncology. Hopefully you can see my mouse, but we've gone from kind of more crude radiation systems in terms of cobalt radiation to moving on to megavoltage radiation, but now we're able to use, this is actually our vault in our machine, more intensity modulated, image guided, hypofractionated radiation that's very targeted in our patients. So in the last probably 5 to 10 years, we have been, as a veterinary radiation oncology community, exploring more and more using stereotactic body radiotherapy to treat various canine cancers. So high dose, high intensity, high precision radiation. And at this point, we're publishing papers to establish baseline outcome data, trying to understand prognostic information in dogs. And from this foundation information, ultimately use this to design and perform more prospective comparative canine oncology trials with more kind of novel or experimental therapeutics in combination. And so just shown here, some of our tumor types that we've been treating with stereotactic radiation and publishing on outcomes. We've treated dogs with soft tissue sarcomas, advanced non-resectable thyroid carcinomas, heart-based tumors. These tend to be chemodectomas or neuroendocrine tumors in dogs, as well as using high-dose radiation, the stereotactic body radiotherapy, as like a limb spare procedure in dogs with osteosarcoma. So one of my interests in that we're working on is trying to study how radiation therapy and the immune response can interact. So radiation therapy kills tumor cells that are directly targeted in the treatment field. And by doing so, radiation can act as an immune stimulus where immune cells are recruited into the tumor microenvironment, activated where they can attack cancer cells in the tumor tissue, but then also kind of throughout the body, if they're kind of primed and targeted to tumor tissue or tumor cells, they can find distant sites, and that's known as the abscopal effect. And there's been evidence in preclinical as well as some clinical evidence as well that these immune responses can be boosted when you combine stereotactic body radiotherapy or more high-dose radiation with immunotherapy to increase anti-tumor immunity and then also increase the incidence of abscopal effects. So in terms of how canine models can play into this field, again, there's interest in the canine model for, excuse me, immune research. Excuse me, one sec. In terms of the general benefits, the dog immune system overall has been well characterized and has some very close homology to humans. They're what we call immunologically experienced as puppies, and kind of throughout life, they undergo, like people do, series of immunizations. They've been exposed to viral and bacterial infections, various environmental exposures. And with this kind of naturally primed immune system, they have developed cancer naturally over time. So that process of the immune system surveillance and ultimately kind of failing and not clearing a tumor that has started to develop over weeks to months and then progress, it's just more of a natural model than injecting tumor cells into a mouse or a rat or a different lab animal model. And so there's ongoing work to try to establish and validate canine cancer models for this translational potential in cancer research. I'm particularly interested in head and neck cancer, where there is a great need for more predictive animal models. Canine head and neck cancer has some biological similarities that could be interesting for this as a translational model. Right up front, this is something that people ask a lot. Dogs, it does not seem that their head and neck cancer is associated with papillomavirus like it can be in people. So this is more of a HPV negative type model. But shown here, this is a CT scan of a dog versus a human patient with more advanced oral head and neck cancer or carcinoma, where dogs, just like people, can have bone invasion. They can have regional and distant metastasis later in the course of disease. Molecularly, they've been characterized to have a number of similarities to people. And again, just to highlight that these are tumors that are developing in dogs with an intact immune system. So we're working to establish canine head and neck cancer as a model for advanced HPV negative head and neck cancer for radiation, immunology, and immunotherapy research. So one of the projects we've completed and we're in the process of submitting is trying to characterize the baseline immune profile of canine oral carcinoma. And so we looked at 32 canine oral carcinoma samples that have been archived in our labs and compared these to normal mucosal samples. And what we looked at just at baseline with the tumor tissue, we did immune profiling with infiltrating immune cells with immunohistochemical staining and then quantification of the cells in the tumor. And what we found is that CD3 positive T cells, which are effector T cells, accounted for the highest mean percentage of tumor infiltrating immune cells, followed by macrophages, regulatory T cells, and then B cells. We also wanted to characterize what we call the microanatomical distribution pattern of T cells. And this has been reported for human patients. Just looking at these patterns, pre-existing immunity, meaning there's already plenty of T cells in the microenvironment, versus immunologically ignorant, so a lack of T cells in the microenvironment, versus being non-inflamed, versus an excluded infiltrate where there's T cells, but they're at the periphery of the tumor and haven't infiltrated into the microenvironment. And so what we found in the oral carcinoma samples from dogs, 77% had that pre-existing immunity distribution pattern, versus fewer were the immunologically ignorant, and then only 2 were the excluded infiltrates pattern. We looked at the immune profile of these tumor samples with respect to gene expression. And so this is gene expression analysis of the tumor samples relative to normal mucosa. And we saw that there was a significant overexpression in the tumor samples of a number of genes which were suggestive of a highly inflamed pre-existing microenvironment filled with more anti-tumor immune cells. But there were also evidence of some other cells as well. So what we wanted to do was actually go through and look at the difference of these tumor samples, those that had high T cell infiltration compared to those with low T cells. And what we saw from the 13 genes that were differentially expressed, the most significantly upregulated gene was CD8. So this is just letting us know that those CD3 T cells that were in the microenvironment are primarily cytotoxic in their phenotype. We also recognize a number of immune checkpoint molecules, so those that are co-stimulatory, like ICOS and CD28, but also some co-inhibitory molecules, which is more consistent with an exhausted phenotype of the immune response, so PD1 and CTLA4. And we also saw that CXCL2 was overexpressed. And this is a chemokine that is known to play a role in recruiting myeloid-derived suppressor cells into the microenvironment. And so the potential for these macrophage-type cells to have immunosuppression. So we looked at the degree of gene expression changes in the high macrophage infiltrating tumor cells compared to those that were low. And we found one gene that was significantly different. It was downregulation of IL-6. So IL-6 is an interleukin that's a pro-inflammatory cytokine. And so seeing that that was down-regulated when we characterized the high macrophage infiltration compared to the low, consistent with those tumor samples having more suppressive effects in their T cell activity. We also went on to calculate a cytolytic activity score, which was a mean of the PERF1 and granzyme A expression for each tumor sample, and then looked at it with respect to the different infiltrating immune cells. And when we saw that there was a very strong correlation with the presence of CD3 positive T cell density in this cytolytic activity score. So again, just characterizing the presence of these CD3 positive T cells with a more anti-tumor immune response pro-inflammatory microenvironment. So just to conclude this study, this is, again, we're working on submitting it, but the first characterization of the immune landscape of canine oral squamous cell carcinoma, where at baseline, these tumors seem to exist in a pro-inflammatory state compared to normal mucosa. But within this sample, there was some heterogeneity that we could investigate further, similar to what's seen in human head and neck cancer. We were able to dichotomize the tumor cells into high versus low infiltration of immune cells. We saw that there was significant changes in gene expression with a pro-inflammatory signature in those tumor samples that had high CD3 T cell infiltration. And those with more macrophages had more of an active suppressive effect on T cell activity. We were able to correlate CD3 infiltration with gene expression in the cytolytic activity score. However, we also saw evidence of immune exhaustion in these samples. Ultimately, we were able to identify some relative immunotherapeutic targets that we could explore in future comparative prospective radioimmunotherapy trials in dogs with head and neck cancer. So I'd like to transition now into one of our other projects, still focusing on dogs with head and neck cancer. Just as some background, again, head and neck cancer, we know it can affect all different regions in human patients, whereas the head and neck cancer of the sinus cavity is not as common in people. it's one of the most common head and neck cancer types that we see in veterinary medicine, so cynonasal tumors. For patients with head and neck cancer, the prognosis is poor, unfortunately for patients with advanced disease. In patients with head and neck cancer that have high risks for regional lymph node metastasis, may have radiation therapy targeted to their primary tumor as well as some of their regional lymph nodes. Ultimately, in order to eradicate any latent metastatic tumor cells. However, the draining lymph nodes are important in generating an immune response. Again, killing off tumor cells in the microenvironment and then priming positive pro-inflammatory immune cells to infiltrate. So if we're irradiating the lymph nodes, ultimately the question is, what's the impact of irradiating lymph nodes on this anti-tumor response, particularly because many immune cells are exquisitely sensitive to radiation exposure. So again, we at Colorado State and in veterinary radiation oncology focus on this comparative aspect of performing our clinical trials. So here's an image of a radiation plan for a human patient with head and neck cancer. But as I mentioned in a translational model for dogs, naturally occurring head and neck cancer with an intact immune system, they can develop head and neck cancer of the oral cavity affecting the lymph nodes. This is a dog with a thyroid carcinoma. What we're going to focus on for this project, though, is head and neck cancer in dogs of the cynonasal cavity. And in our world, you know, we also are in situations where we may be treating regional lymph node irradiation, either for diseased lymph nodes, obviously to target those tumor cells, but also prophylactically and electively to try to clear residual microscopic metastatic disease. There's no standard in vet medicine in terms of when we do or don't irradiate lymph nodes, so we have some room to explore this with our patients. We can treat our patients, and we sure do, with conventionally fractionated radiation protocols, but again, we are doing more kind of exploratory work with stereotactic body radiotherapy in dogs. So just some background on cynonasal cancer. As I mentioned, it's a smaller subgroup of head and neck cancer in people. About 0.5 to 2.5 cases per 100,000 people has been reported. The most common are epithelial tumors or carcinomas. That's been reported to be greater than 80% of all cynonasal tumors. In dogs, like I said, also get cynonasal cancer with a reported higher incidence, about 15 cases per 100,000 dogs. Just like in people, cynonasal carcinoma is the most common tumor type, 60 to 80% of tumors. Again, we use radiation therapy actually predominantly for both local and regional tumor control with either conventionally fractionated or more often now, it seems, stereotactic body radiotherapy. So in this study, our objective was to investigate the impact of regional lymph node irradiation on the immune response in a trial of dogs with cynonasal cancer undergoing SBRT. Our hypothesis was that regional lymph node irradiation would reduce local lymphocyte populations in the tumor microenvironment, as well as regional lymph nodes following SBRT, compared to dogs treated with SBRT targeted just to their cynonasal cancer alone. So for our study design, this was a prospective randomized trial. Dogs entered into one of two treatment groups, SBRT to their nasal tumor alone, or SBRT to their tumor and their regional lymph nodes. Our protocol was 30 gray, delivered in 3 consecutive daily fractions of 10 gray, and this was administered with VMAT and 6 MV photons. Pre-treatment, every dog had a CT scan as well as staging diagnostic tests to confirm that they had no evidence of disease outside of their nasal cavity. Again, 3 fractions of 10 gray was administered. Whoops. All right, and then this is a slide for what our sampling was throughout this trial. In order for us to evaluate the local immune response, we developed kind of a new model for immune monitoring of cynonasal cancer, where what we did is we flushed saline into their nasal cavity. All the dogs are anesthetized for radiation treatment. So while they were asleep, using a nice soft catheter, as well as nice warm saline, we flushed saline into their nose and then collected all the cells and protein from their nasal cavity, and then analyzed the cells and proteins up in the lab. We also, we did this pre-treatment and then with every fraction of radiation. And then 2 weeks later, we repeated we call it nasal lavage again, as well as we removed one of their mandibular lymph nodes, ipsilateral, so on the same side as their tumor, to analyze the lymph nodes that were either irradiated or spared. This is a distribution of the patients in the trial. So a variety of different breeds and ages. We had mostly carcinomas, but we did have a couple sarcomas. Some tumors in the population that we have is inconclusive with biopsy, just we can't get a solid sample to make a clear diagnosis. So at this point for this trial, we included dogs where we knew it was a nasal tumor, but we couldn't characterize exactly what kind. And so this is a distribution of the flow cytometry data that we found. So the blue bars are dogs treated with SBRT to their nasal tumor alone, versus the yellow are SBRT and their lymph nodes irradiated. So the first set of bars, this is pre-irradiation. These are cytotoxic T cells versus helper T cells versus activated B cells. And what we found is this is on the day 3 of radiation therapy. So they were radiated after the treatment. We collected cells from their nasal cavity. And we saw that there was a significant reduction of cytotoxic T cells, helper T cells, and activated B cells from the dogs where the lymph nodes were irradiated at the time of their primary tumor irradiation. And then we also looked at cytokine levels from the nasal lavage fluid that was collected. So this is dogs where their lymph nodes weren't irradiated. And we saw from day 1 through to day 3, and then this is at 2 weeks, there was a a relative spike in cytokine release that was quantified from their nasal lavage fluid. But when the lymph nodes were irradiated, that effect was dampened in the dogs of this trial. And then we also looked at the lymph nodes that were irradiated. This is CD3 staining versus, these are T cells, versus that same CD204 for macrophages. When you spared lymph nodes from radiation, you had plenty of T cells and very few macrophages. But when the lymph nodes were irradiated, we had depletion of those T cells and then an increase in macrophages in the irradiated lymph nodes. Again, this is 2 weeks after radiation. And then this is quantification here, where there's a significant increase in the CD204 macrophages in the irradiated lymph nodes compared to those that were spared. And then with respect to gene expression, these are genes in the irradiated cells, irradiated lymph nodes compared to non-irradiated. There was an increased expression in DNA damage repair genes, as well as interferon gamma signaling, which makes sense for irradiated tissue. And sorry, I forgot to mention, this is using a nanostring 360 immune panel that we use for our gene expression analysis. And then in terms of decreased gene expression from the irradiated lymph nodes, there is a significant reduction in pro-inflammatory immune response associated genes, tyrosine kinase receptors and activity, as well as cell-cell adhesion. So we found from this kind of preliminary trial is regional lymph node irradiation in dogs with cynonasal cancer reduce the percentage of local pro-inflammatory immune cells as well as local cytokine levels. It reduced the lymphocyte density in the irradiated regional lymph nodes, as well as increased populations of macrophages and regulatory T cells in the irradiated lymph nodes. With gene expression analysis, radiation of the lymph nodes had an increase in DNA damage repair and interferon gamma signaling, and decreased expression of genes associated with pro-inflammatory immune responses, tyrosine kinase receptors, and activity in cell-cell adhesion. So this is an ongoing project. We're working with collaborators to study these effects in mice, as well as archived human samples, to try to understand what's the clinical impact of regional lymph node irradiation on anti-tumor immunity. So what this study has really prompted us to do is explore more this idea of using dogs with cynonasal carcinoma as a model for the human form of disease, since it is more rare in people and much more common in dogs. And we're, following with radiation protocols with the SBRT that could be applied for human patients. Where can we bridge the gap for this disease? We did similar to what we did for the oral carcinomas. We're starting to evaluate archived cynonasal carcinomas in dogs. And we did the same immune profiling with immunohistochemistry to evaluate the percent of infiltrating immune cells at baseline. And what we found is that in this tumor type, macrophages was the predominant infiltrating immune cell. So now we're trying to look at how immune responses to radiation with a combined immunotherapy protocol where it's more myeloid cell targeted, so going after these macrophages, which may be immunosuppressive, can hopefully improve local tumor responses. And I'll go into a little bit more in a couple slides of what the mechanism is, but we're looking at propranolol and losartan as our combination immunomodulation experimental group. So our hypothesis is that these repurposed immune modulating drugs will deplete the immune suppressive myeloid cells from the tumor microenvironment, ultimately restoring positive anti-tumor immunity, both local and systemically, and then leading to more durable radiation local tumor responses. So this is a project that's underway. I'm just keeping an eye on the time. I'm working with collaborators at the University of Colorado. This is through, there's a head and neck spore within their group, and I have a career enhancement award to do this project, where we're looking at combining these immunomodulatory drugs with radiation in dogs. So again, as radiation recruits immune cells to the microenvironment, particularly SBRT being more highly immunogenic than some of the lower doses of radiation, we want to evaluate how we can kind of boost the more positive anti-tumor immune responses and overcome those immunosuppressive influences that continue to exist within the tumor microenvironment. So we're looking at losartan and propranolol. Losartan is an angiotensin II receptor blocker, a CCR2 antagonist. What it does is it decreases monocyte recruitment and leads to depletion of tumor-associated macrophages in the microenvironment. And then propranolol in combination, this is a beta-adrenergic receptor antagonist, blocks adrenergic stress associated with tumor immunosuppression, reduces immunosuppression of myeloid-derived suppressor cells, and then therefore increases the ability of cytotoxic CD8 positive T cells to function in a positive way and for anti-tumor immune responses. So our hypothesis with this project is that this TME, or tumor microenvironment modifying immunotherapy with losartan and propranolol, will deplete the myeloid-derived suppressor cells and tumor-infiltrating macrophages, restore T cell immunity in cynonasal cancer treated with SBRT, leading to improved responses. We are going to be doing this with an orthotopic murine head and neck cancer model, as well as a naturally occurring canine cynonasal cancer to investigate our hypothesis. So with the mouse model, we're again working with our collaborators at CU, where they have a number of really interesting orthotopic head and neck cancer models that they grow in mice that are immunocompetent. This is a tumor model that is oral cavity associated, so injecting into the buccal mucosa of mice. We have plans to do this with this model, looking at the different treatment groups, but we're also considering implanting these cell lines into the maxillary sinus of the nasal cavity of mice and evaluating this as a cynonasal carcinoma model with a lot of kind of quotes around it. However, we recently, just this week, and I'm really excited about it, have kind of identified there's a rat model of syngeneic cynonasal carcinoma where we can try something similar. So it's an actual cynonasal cancer cell line that we can implant into the nasal cavity, hopefully, of rats and do more rodent-based research so we can do more mechanism studies. In terms of our trial for dogs, this is ongoing. It's prospective, randomized, 2-arm controlled canine clinical trial where the treatment groups are SBRT alone with that same 10 grade times 3 protocol, or SBRT with propranolol and losartan. So dogs, if they're in the propranolol and losartan group, they have 1 week of the drugs prior to radiation. Just as before, pre-treatment, we make sure that everybody is has disease localized just to their nasal cavity, no evidence of metastatic disease. Prior to treatment, they have another one of those nasal lavage procedures. We're doing blood sampling to evaluate systemic effects. They have 3 fractions of the radiation on the last treatment. Again, collect nasal lavage and blood, have them back 2 weeks later for their nasal lavage blood. And then at this time point, we get another tumor biopsy. And then 3 months later, we're having them come back in for another CT scan to evaluate local response, as well as get additional sampling. And then in terms of the analysis that we're performing from the tumor biopsy pre and post treatment, oh, I've got my thing kind of in the way. That's okay. We're doing formalin fixed tissues so we can do, again, that immunohistochemistry evaluation of infiltrating immune cells. We're also freezing samples so we can do RNA sequencing, nanostring evaluation. From the blood cells in the serum, we are evaluating PBMCs as well as that same cytokine analysis from blood samples, doing it again, cytokine analysis from the nasal lavage samples, as well as looking at flow cytometry for the distribution of the immune cells over time throughout the trial. And we've also started doing some single cell RNA sequencing from these nasal samples, so excited to be doing some with that technique as well. So in terms of ongoing work, we are determining the extent of similarities or differences, what might be interesting there between human and canine cynonasal carcinoma samples. So that's underway. Like I said, we're trying to determine the impact of combining SBRT with these propranolol and losartan drugs. We've also just started, we have 1 growing, and we're hoping to do more, developing canine cynonasal carcinoma cell lines for in vitro studies. And then, like I mentioned, do more with the single cell sequencing of cells collected from the nasal lavage samples. All right, again, just keeping an eye on time. I think we're okay. But you guys let me know if you want me to stop here. This is just another project I wanted to highlight. This is, again, looking at radiation and immunotherapy combinations for dogs with spontaneous tumors. This was just recently published in a special issue on molecular radiobiology in the International Journal of Molecular Sciences. I just have the reference there if you wanted to check it out. I will go through this quickly again in the interest of time. Radiation itself, high doses can be immunogenic. In this experiment, we were trying to look at a different combination of immunotherapy. So a lot of immunotherapy that's being investigated is checkpoint inhibition, where you're taking the brakes off the tumor immune suppression, antibodies against CTLA4, PD-1, PD-L1. But there's evidence where, unfortunately, there are a number of patients that are either resistant to this immunotherapy up front, or if they have a nice response, go on to relapse. So just evaluating different immunotherapy options. So the alternative, kind of what we call stepping on the gas to stimulate anti-tumor immune responses. So we evaluated agonistic anti-OX40 antibody in combination with a toll-like receptor 9 ligand. So in terms of our study, we wanted to investigate immune effects of combining SBRT with local injections of OX40 antibody and TLR, it's actually 3 and 9 ligand in a spontaneous canine cancer model. Our hypothesis being that the combination would enhance anti-tumor immune responses compared to dogs treated with SBRT alone. So here is our trial design, similar kind of before in terms of we evaluate everybody pre-treatment to make sure we have an understanding of the tumor extent. We had tumor biopsies as well as serum collected. We also evaluated tumor oxygenation in this study. Vasculature oxygenation, there's evidence that that can affect immune responses, so we wanted to look at that as well. These were various tumor types, so they were treated either with a 3 or 5 fraction SBRT protocol, and then 2 weeks after radiation came back for tumor biopsy, blood collection, and another measurement of tumor oxygenation. And so this is just images of one of the dogs in the study with an oral tumor. These stars indicate, we made like injection maps to try to administer the immunotherapy as homogeneously as possible, versus dogs in the radiation alone group had injections just with saline. Here's a distribution of the dogs in the study. So again, various breeds, tumor types. It was a mix of oral malignant melanoma. Mucosal melanoma is pretty common as an oral tumor in dogs. Carcinomas as well as soft tissue sarcomas, and they were distributed evenly throughout the groups. This is another quantification using immunohistochemistry of infiltrating immune cells. And this is 2 weeks after radiation. And so what we found, this was interesting to us, in the dogs treated with SBRT and immunotherapy, there was a significant reduction in CD3 T cells, but also a reduction in regulatory T cells, this FOXP3, which are known to be immunosuppressive. We also looked at gene expression analysis in these dogs, and this was relative to the 2-week post-treatment gene expression values, and we looked at a number of different panels, those associated with signatures of regulatory T cells, where we saw dogs treated with immunotherapy had a reduction in FOXP3. Those genes associated with an exhausted T cell signature. We saw SBRT and immunotherapy treated dogs had a reduction in their expression of CTLA4. With myeloid cells, again, that macrophage type immunosuppressive phenotype, we saw immunotherapy reduce the levels of IL-8 expression. There wasn't any significant differences with gene expression with respect to effector T cell associated genes. We looked at serum cytokine levels in these dogs, and we found that there was overall an increase in cytokine levels in dogs treated with SBRT and immunotherapy. The one that was significantly different was IL-7. And then again, we mentioned that we looked at tumor vascularity and oxygenation changes. We did this with optical spectroscopy device on the Xenoscope system, where you place a probe on the tumor and get a number of measurements at different time or different places on the tumor, as well as serial time points. And we correlated that with CD31 immunohistochemistry. And what we saw, this wasn't significantly different from the groups, but there was a trend that was consistent with those treated with radiation and immunotherapy had reduced kind of microvasculature as well as reduced hemoglobin concentration, kind of from the pre-treatment to the post-treatment values. And ultimately, we were not quite sure what this means, but just something that we found in this trial that we can evaluate further. So summary or conclusions from this trial, we found that dogs treated with radiation and immunotherapy had decreased infiltration and influence of the immunosuppressive cells in the tumor microenvironment. They had decreased intratumoral CD3 positive T cells, which again was interesting, but they had a significantly increased level of pro-inflammatory cytokine IL-7 in the serum samples. Overall, this was safe and well tolerated in the dogs, but it wasn't designed for this, but we didn't find an effect on progression-free or overall survival times. Doing this in a larger trial, we would be increasing the dosing of the immunotherapy as well as controlling for tumor type and stage of disease at the time of treatment. And then again, I might skip through this just so we have time for questions, but I've included just some things that considering expanding on canine translational radiation research, just ongoing work to try to establish and validate new models, recognizing what are the similarities and differences between dogs and human cancers that could be explored or exploited to understand more of the biology and treatment responses. Also understanding that normal tissues and health following radiation can be evaluated in dogs. We're trying to always develop new tools, obtain new access to new drugs or devices, and very much open to collaborations and trying to integrate canine cancer research for, again, the greater good of improving cancer outcomes for all species. I include here just some resources in terms of what's available now for integrating into comparative oncology research. This is now a couple years old, but a nice collection in Frontiers in Oncology. There's a whole featured issue on emerging translational opportunities in comparative oncology. So if you wanted to check that out. And yeah, again, thank you so much for the opportunity to speak with you today. I want to list my funding and as well the team that I'm very fortunate to work with every day. They're a good group and I'm lucky to be here. So thanks. And I'd be happy to take any questions.